Ladies and gentlemen, I wish you all good morning and very welcome to Nobel Forum for the announcement of this year's Nobel Prize in Physiology or Medicine. So I will first read the announcement in Swedish, followed by English, and we will then present some background to the prize and then open up for questions, of course. Nobelförsamlingen vid Karolinska institutet har idag beslutat att Nobelpriset i fysiologi eller medicin år 2018 skall delas lika mellan James P. Allison och Tasuku Honjo för deras upptäckt av cancerbehandling genom hämning av immunförsvarets bromsmekanismer. The Nobel Assembly at Karolinska Institutet has today decided to award the 2018 Nobel Prize in Physiology or Medicine jointly to James P. Allison and Tasuku Honjo for their discovery of cancer therapy by inhibition of negative immune regulation. Here we see the two new laureates. James P. Allison was born in Alice in Texas in the United States. He performed his prize-winning studies at University of California, Berkeley, and at Memorial Sloan Kettering Cancer Center in New York. He is now active at the University of Texas MD Anderson Cancer Center in Houston. Tasuku Honjo was born in Kyoto, in Japan. He performed his prize-winning studies at Kyoto University, where he's still active. I will now leave the word to Professor Claes Kärre, member of the Nobel Committee, who will describe this year's discovery. So please, Claes.
Ladies and gentlemen, let us start by a summarizing metaphor. It is all about interference with the brakes of our immune system as a means to unleash this defense and harness it to develop treatment of cancer. Cancer is a common term for many different diseases caused by uncontrolled proliferation and migration of cells. Eventually, cancer cells can spread to distant organs, so-called metastases, usually leading to death. Cancer represents a major medical challenge in the global perspective. There are three pillars in the treatment of cancer: remove it by surgery, eliminate it by radiation, or attack it with drugs that harm the cancer cells. Treatment has improved considerably during the last 50 years. More than 2 out of 3 are cured today. But note that these are average figures which conceal major differences between cancers in different organs, between early and advanced disease, and also between different countries. For many cancers, very few patients can be cured. Every year, many millions of people die from cancer. So novel types of treatment are desperately needed. There has been a huge investment in basic research with the aim to acquire knowledge as a platform for new therapies. One area was of particular interest: can immunity be harnessed to attack cancer? So let us look at our immune system. It protects us from infections. It comprises a great diversity of cells and molecules with different functions, and they can be found all over the body. A fundamental property of the system is its ability to discriminate between self and non-self, so that viruses and bacteria can be attacked. And indeed, innovative basic research in tumor immunology during the last 50 years have revealed a number of ways whereby cancer cells can also be recognized as non-self. This led to cure of cancer in many mice and also to successful treatment of some patients. However, it was impossible to develop efficient, generalizable therapies for clinical practice. Clearly, an important piece of the puzzle was missing, and this is what our laureates this year have solved. Now, let us take a closer look at the T cell with the T appearing here, and we focus on it. In the 80s, scientists had discovered T-cell receptors that bind to structures recognized as non-self. You see the structure as a dot in purple here. These structures can be picked up from cancer cells and be presented by so-called antigen-presenting cells. T cells that recognize something foreign with the receptor, as here, they get a signal to act, but this is not sufficient to get them going. Additional proteins on the surface of the T cells must also engage in the decision, acting as accelerator or gas pedal. They were called co-receptors, and the research field around them was termed co-stimulation. Many researchers contributed and observed that even more molecules were involved, but with quite the opposite function. They instead worked like brakes. And here they are in yellow, or at least one of them in yellow. And this one is called CTLA4, a complicated name, but please remember it. It will be very important. So when you pull the brake, it tells the T cell to hold back, stop. Allison was one of the researchers who observed that this molecule works like a brake. You can see that it interferes with the accelerator on the T cell. Now, some research teams started to explore this to treat autoimmunity, but Allison, he had an entirely different idea. He had developed an antibody to CTLA4, shown in green here. And if you block or release the brake with the green antibody, Allison reasoned, the consequence should be that the T cells are unleashed and become more active, and they should now hopefully attack the cancer cells. This was the hypothesis, and the key experiment was performed in Allison's laboratory at University of California, Berkeley, over the Christmas in 1994. And the results were spectacular. The mice who had been treated with the antibody, the yellow curve, were cured from cancer, while the control mice developed large tumors. This seminal study represents the birth of a new concept for immunotherapy, today often referred to as immune checkpoint inhibition. The brake acts in the checkpoint. Allison was now eager to apply it for benefit of patients. At first, there was only limited interest, but eventually a small biotech company took on the challenge to produce the human antibody, the first step on a long journey. A few years later, the first clinical trials were started, and one comprised 14 patients suffering from metastatic melanoma, a pigmented cancer of the skin. And this picture shows how a metastatic melanoma can present on the patient. Now, he started to treat the patients, and some of them showed striking responses. The tumors shrunk, they could even disappear. Other patients showed a more modest or no response at all. And severe side effects were noted in some patients, but the results were positive enough. And this started a long series of larger trials conducted by clinical colleagues. To cut a long story short, in 2011, the treatment was approved for inoperable metastatic melanoma. Allison had now established a novel concept for cancer therapy, but several questions remained. Could checkpoint therapy be developed to cure more patients, and could it be generalized to other types of cancer? Studies that would lead in this direction were already ongoing in a laboratory across the Pacific in Japan. Actually, they had been started several years before Allison's key discovery. So we cross the Pacific, we go to Japan, and we go back in time to the point where Allison had started. And you will remember and recognize the T-cell receptor and the accelerator. Enter our second laureate, Tasuku Honjo, who in 1992 discovered a new protein on T cells, which he called PD-1. And PD-1 is shown on yellow here on the T cells. He did not understand what the molecule was doing, but together with his colleagues at Kyoto University, he launched an ambitious, curiosity-driven research program to understand its function. Several years of research later, he could reveal a chain of molecular interactions, leading him to conclude that PD-1 also acted as a brake, just as CTLA-4, but in a different manner. You step on the brake, and the T cells holds back. In addition, Honjo had identified the ligand, the molecular partner of PD-1, and he found that it was expressed on antigen-presenting cells, but also on cancer cells. And now it became immediately evident to test whether blockade of PD-1 could also be used to unleash T cell responses against cancer. So what happens when you release the brake like this? Honjo's and other groups immediately started to gather evidence for this. And here we are looking at one of the studies of melanoma, the skin cancer, and in this case it is spreading to the liver in mice. The control mice on your left developed multiple metastases in their livers, while several of the mice who had been treated with the antibody to your right had almost no metastases at all. In this study, Honjo correctly predicted that anti-PD-1 treatment might complement the anti-CTLA-4 treatment, since the two worked via different mechanisms. He also predicted that PD-1 might give less side effects. Honjo now also turned to the pharmaceutical industry. An intensive program started, conducted by other clinical scientists, mainly in the US. Again, similar or even more dramatic responses were noted, not only in melanoma patients. And FDA approved the anti-PD-1 treatment for melanoma in 2014 and for lung cancer as well as kidney cancer in 2015. So now we have two treatments available for patients based on immune checkpoint therapy, and today we have even more. And it's time, I think, to look at some results in patients. These CT scans are from a patient treated with anti-PD-1, but they illustrate processes seen also with anti-CTLA-4 treatment. You can see the tumor marked by the red arrow in the lung. Note how it has shrunk dramatically to your right, and it's on its way to disappear at 4 months after initiation of the treatment. But please note also what happened shortly after the treatment had started, at 2 months in the middle. The tumor appears to be significantly larger than it was from the beginning, but this is not real growth of the cancer. It is due to swelling caused by the inflammatory response, with many immune cells entering the tumor. And this is called pseudoprogression and is observed quite frequently, and it caused confusion initially, since other types of cancer treatment usually results in an immediate response and shrinkage of the tumor. Let us finally look at an example of compiled results from many patients. These curves represent survival of treated patients with a certain type of melanoma. And you should note that just a decade ago, most patients with advanced disease like this would have died already after 2 years. Previous standard therapies resulted in long-term survival for few, 10% or less, and it would have looked like this. The curve compares 3 groups of patients treated with checkpoint inhibition. The release of one brake, CTLA-4 in green, release of the other brake, PD-1 in blue, and release of both in yellow. And we can appreciate that both treatments are working, anti-PD-1 in a higher proportion of patients than anti-CTLA-4, but please note the combination, where more than 60% of the patients are still alive after 3 years. This is a spectacular advance in the treatment of a solid metastatic tumor, and we can expect even better results in the future when these different checkpoint inhibitors will be combined with conventional treatments. So, in summary, Allison's and Honjo's discoveries have added a new pillar in cancer therapy. It represents a completely new principle because, unlike the previous strategies, it is not based on targeting the cancer cells, but rather the brakes, the checkpoints of the host immune system. The seminal discoveries by the 2 laureates constitute a paradigmatic shift and a landmark in the fight against cancer.
Thank you very much. So if we could have the lights a little brighter here. So I would like to open up for questions. I would like to mention that to our help here we have some distinguished experts, all from the Nobel Assembly and the Nobel Committee. Dr. Jonas Berg, who is an expert in oncology, Ted Smith from the Nobel Committee, Dr. Anna Waddell, the chair of the Nobel Committee, and of course Klaus. So please, if there are any questions, here we have one.
Hello, my name is Fred from China's National Xinhua News Agency. I've got a question for the experts. So this year's Medicine Awards is given to the new cancer treatment. So what are the significance of this award? Are we saying that the cancer treatment has been a huge challenge nowadays for the human being? What are the significance for this award this year?
Ted?
So this type of cancer treatment is very interesting because it works in a completely new fashion. All previous cancer treatments were essentially directed against the tumor cell, but this particular treatment is directed against the immune system. And one interesting aspect is that it works on many different types of tumors, not only on one particular tumor, but on many different types.
Here we have one question. Can you please wait for the microphone? Okay, we'll start with one up here and then your. Okay. Please go ahead.
Okay, I have a question about James Allison anti-CAR-T4 therapy. I know that another excellent scientist from Yale University, Lipin Chen, he's also a highly eligible candidate for this. Just would like to know the reason that he's not being selected. Thank you.
Klaus, do you want to respond to that?
The question was about another scientist who is not given this year's award. Now, let me first say that the Nobel Committee divides the world, or the people of the world, in two groups. The ones that have received the Nobel Prize in Medicine and the ones who have not yet received it. And we love to comment on the ones who have received it, but prefer to refrain from comments on the others. Having said that, I would like to add that, as I tried to imply in this presentation, many scientists contributed to this whole field. And if you want to read about the contributions of different scientists, many of them very, very smart and clever, who contributed a lot, we refer you to the advanced information that is, I think is now out on the Nobel web, where you can read about all the contributions of many other scientists, which were also important, but these two stand out.
Okay, here's one question.
Yeah. Thank you. I'm Xifei Chen Axel Song from Green Post and China Radio. I'd just like to ask you that when we talk about this immune checkpoint therapy, does it mean that it is a method that narrowed down the point to check for the cancer treatment, or how better it is than the previous ones? Thank you.
Could I ask Jonas Berg to answer that question?
The effects of these checkpoint inhibitors are quite remarkable because they really define a new concept where you release the brakes. Previous types of immune therapy, they were really trying to make use of the immune system by stimulating it and enhancing its capacity. But here, in that concept, you didn't release the brakes, and for that reason those therapies were not as successful as these therapies. And the fundamental thing also is that these therapies, as we are discussing today, they are apparently and very nicely working in previously quite therapy-resistant tumors, as mentioned here, malignant melanoma, different types of lung cancer, to some extent renal cancer, to give some examples. while quote-unquote simpler cancer, which have been sensitive to endocrine therapy, cytostatics, radiotherapy, etc., the effect by the conventional therapy has been quite good, but for the tumors I mentioned, that present armamentarium up until today have not been that fantastic.
Yeah, what about the side effects?
Could maybe, who wants to answer that? Maybe it's best that you go ahead with that, Jonas.
The side effects are there, it's correct, but if you compare with other approved drugs or therapies for cancer, there are by all means always side effects. And if I compare the armamentarium, the new drugs we are discussing, the checkpoint inhibitors, the side effects are for almost all patients manageable, although they are in a scale up to 5. They are not rare with grade 3 to 4 side effects, but mostly of them, they are reversible. And it's clearly so if you look at the benefit, namely the effect on the cancer, and then you balance that versus the risk, there is no question that there is a clearly positive benefit-risk balance for these, for the therapy of the cancers we have discussed here.
There's one up here.
This is from Nordic Chinese Times. I have a question for I would like to thank you for these two great scientists. But we have now run thousands of clinical trials with PD-1 and CTLA-4 and with combination therapy. So we even run out of patients. So what's the next future or rational design for this combination with this checkpoint therapy? Thank you.
Ask Anna to respond to that.
Yes, I think what's most promising now, we have now a completely new tool to add to the toolbox. So it has sort of revitalized the whole area. So to find the right combination for the right subgroups and even for the right patients and go into a more individualized therapy will be a very important process. There are very many studies, of course, ongoing doing that. So there will be more concepts, more checkpoint inhibitors most likely, but combining what we already have also with conventional therapy, there's a huge benefit to gain there, I'm sure.
Do you want to add something?
Yes, I can add that this is a very active tool. So let's take it last Friday, 2028. FDA approved yet another anti-PD-1 antibody, which is called inhibition. I can also add when it comes to the side effects, the adverse effects, that there are differences. So the latest drugs directed against PD-1 and PD-L1, the ligand and PD-1, have fewer side effects than the first candidates that came up. Those directed against anti-CTLA-4.
The powerfulness by combined therapy, I can mention triple-negative breast cancer in a small randomized phase 2 study. If you just give chemotherapy in 20%, 1 in 5 patients, the tumor totally disappeared in the areas which were there before therapy. If you do the combination of chemotherapy plus a checkpoint inhibitor, in 6 out of 10, 2 out of 3 patients in reality, the tumor disappeared. So it was a dramatic improvement from 20 to 60% in this very early study, demonstrating that combination for some patients could be quite useful using the conventional therapy strategies.
Mr. Perlmann, David Keaton from the Associated Press. This is a question for you. We saw earlier a photo of you speaking on the phone to the winners. Could you tell us a little bit how they reacted when they heard the news that they were this year's winners?
Yeah, well, what you did see was speaking on the phone with Dr. Honjo. So I didn't get in touch yet with Dr. Allison. He might know it now, but I never got hold of him. Dr. Honjo sounded extremely pleased. I have a little bit difficult time to guess if he had some expectancy of this news, but he seemed rather surprised, and he expressed very clearly how satisfied he was, also of sharing the prize with Dr. Allison. Are there any other questions? So with that, I think then, if there is no one else who want to ask anything, that we can conclude this press conference. And there are some of you who asked for interviews, so we will try and satisfy all requests as quickly as we can. So thank you very much for coming. Thanks.
Tá longe, só quando a gente já tiver velho. Mas você vê, quando que eles começaram a fazer? É. Foi pra ser recente, né? Mas quando que eles começaram a fazer? Ah, os papers são mais ou menos recentes. Você tem lá 4.996.
Professor Claes Kärre, member of the Nobel Committee. We have just heard that this year you're awarding the discovery of checkpoint inhibitors. Could you please explain what this is?
Well, first of all, this is a treatment against cancer, and it's a new principle for treatment because unlike previous treatments where you target the cancer cell, this treatment targets the host's immune system, the patient's immune system. And it does so by releasing or blocking the brakes of the immune system. And when you release the brakes, things go faster, and we have a more strong response against the patient's cancer cells.
At least one of the laureates' names, James Allison, he has been mentioned for quite a while. How come you are awarded the prize this year? Why now?
Time is ripe. The first approved drug based on this treatment came in 2011, and patients have been treated for several years now, and we can see the long-term outcome, and it's very convincing, and we consider it really a big step in the fight against cancer.
It was quite complicated, different cells and different receptors, but in order to describe this prize in an accessible way, what kind of metaphor would you use?
I would like to use the brake metaphor, the brake pedal that you have on a car, for example, where you can step on it and everything stops, and you can release it and you get things going. That's the best metaphor.
This is a new kind of cancer treatment. What cancer patients can be treated using this method? Are there any limits?
The most dramatic responses are seen with a particular type of skin cancer, malignant melanoma. And I'm sure most viewers know what this is. It's a pigmented cancer on the skin. Other cancers that respond at least to PD-1 treatment are lung cancer and kidney cancer and lymphoma and many other cancers actually also. So it will be very exciting to see in the future exactly how broadly this can be applied.
Personally, what makes you enthusiastic by this prize?
I myself am an immunologist, and I have actually worked in tumor immunology for many, many years. And there has been a lot of interesting new discoveries in tumor immunology where we revealed basic mechanisms, but they were never sufficient to be able to cure cancer in patients. They were sufficient to cure cancer in mice, which is somewhat easier. And all the other previous attempts to use the immune system, such as vaccination or giving cells, they worked in some patients, but you could never generalize it to many patients. And this is what is different about this one. And it makes a big difference for certain patients. Some patients survive and are cured, and some patients get 4 years rather than 1 year, and that is quite important.
Now, if we turn to the laureates, James Allison and Tasuku Honjo, who are they?
I have met them, but I don't know either of them very well. But Professor Allison is a basic immunologist. He's not trained in medicine, but in biology from the beginning. And he became interested already when he was a small boy, and his father gave him a chemistry kit. And he has had a splendid career in basic immunology. He made many important discoveries before this one, and he has been in many different centers in the United States. Berkeley was mentioned here, but he has also been at the Memorial Sloan Kettering Cancer Center in New York and in MD Anderson in Texas, where he is now. I'm also told that he plays the harmonica in a blues band called the Checkmates. Professor Honjo is also a very distinguished basic immunologist. He's actually an MD, so he has medicine training, but he has also contributed very important discoveries in basic immunology before this one, and he was not in cancer immunology from the beginning, similar to Jim Allison, who also was not in cancer immunology. But these basic scientists made their discoveries and persistently followed them through and developed these fantastic treatments then. I'm not sure whether Professor Honjo plays any instrument, but I know that he plays golf and is very interested in art.
When they were doing this discovery, were they looking for this, or was it a surprise for them when they found it?
For Professor Allison, I think he was working with the basic regulation of the immune system, like many others, and he did not discover the molecule that he was working on, but he realized its potential. So he rapidly tested an idea that nobody else tested. I don't know how expected it was for him to get into cancer, but he did. Professor Honjo, I'm sure, he did not think of cancer at all in the beginning. When he looked and found his molecules, he was doing completely different things and actually probably had ideas that it was doing something completely different. But as he went on and he gradually revealed how this molecule worked, he understood with inspiration from Allison how this one could add and even become even more efficient.
And finally, if you were to do an elevator pitch on this prize with the goal to simply convey the overall concept in an exciting way, what would you say then?
These scientists have discovered how you can block the brakes of the immune system so that it goes much faster and acts in a much more intensive way, and thus can attack cancer cells, so we can cure cancer with it.
Excellent. Thank you very much.
Thank you.